Hello and welcome to this Revision Monkey video on what's going to be in the 2022 exams. This video is for the AQA specification and it's for combined scientists, and that's the trilogy version of combined science, which means you study 6 papers at the end of your course, all of which are 1 hour and 15 minutes long. This video covers the major focus content for biology paper 2, and it's for the foundation tier. Do keep an eye out in the description because I'll put a link there to a video that just focuses on the required practical that they've told you to focus on for this paper, and I'll also put a link there for some questions a little bit later on. So first of all, looking at what's not going to be assessed in this paper, actually there's quite a few things that they've taken out of this exam, and these are the human nervous system, hormones in human reproduction, contraception, sexual and asexual reproduction, meiosis, sex determination, variation, evolution, selective breeding, extinction, resistant bacteria, adaptations, biodiversity, land use, deforestation, global warming, and maintaining biodiversity. They've told you the required practical to focus on, and this is organism distribution, and as I said, there'll be a link in the description that focuses just on that required practical, so do look at that video. But the rest of this video will look at the content that the exam board have told us will be the major focus of the exam. And these topics are hormonal control in humans, reproduction, adaptations, interdependence and competition, and organisation of an ecosystem. So that's what's going to be the focus of this video, and of course I've removed anything that they've said won't be included in the exam. For example, in that 4.7.1 adaptations topic, they've actually told you to ignore the adaptations part and just focus on the other sections of that part of the topic. So the rest of the video will be on this. Do feel free to take notes as you go along, and also look out in the description for a link for some questions as well. The endocrine system. It's a really difficult word just to describe glands that produce hormones. Controlling our body by hormones is slower than that of nerves, than a nerve response. It acts in a more general way. And usually the response lasts longer. Now there are several endocrine glands that you need to be aware of. The most important one here in your head is called the pituitary gland, and that has got a little bit of a nickname called the master gland because that is often responsible for switching on and off other endocrine glands in your body. The one in your throat here is called the thyroid gland. That produces a hormone called thyroxine. The pituitary gland produces lots of hormones that we'll talk about later as well. The adrenal glands here, they produce adrenaline. You've got the pancreas here, a really important gland that produces lots of different hormones, and we'll talk about the pancreas later on too. And depending on whether you're male or female, females have ovaries, and we'll talk about those when we talk about the menstrual cycle, and the men have testes, and the hormone that's produced there is testosterone. We're going to look now at the pancreas in a little bit more detail because we're going to talk about one of the things that's important in homeostasis, which is controlling the blood sugar levels. So the pancreas is involved in controlling blood sugar levels because it produces a hormone called insulin. It's something that your bodies will produce all of the time. And what the insulin does is, if there's too much glucose in your blood, I'll just represent the glucose by a G. If there's too much glucose in your blood, the pancreas will start producing more insulin, and that will cause the glucose to go into your muscle and your liver cells. And here it can be stored as glycogen. So the liver itself stores glucose as glycogen. So difficult words here. Glucose and glycogen sound very similar, but if the blood glucose levels are too high, it is controlled by this hormone, insulin. And the trouble with controlling blood glucose is it is linked to a disorder called diabetes. There are two types. Type 1 diabetes is the type that is inherited. And in this type of diabetes, your body produces little or no insulin. And this is the type of diabetes that the doctor will prescribe insulin therapy. So that involves insulin injections. And you have to keep a close monitor of the glucose intake that you're taking into your body. If your blood glucose gets really low, you will quickly eat some biscuits or some foods that are high in glucose. And you also need to regularly remember to go and get your insulin injections. Type 2 diabetes, however, is caused by lifestyle factors. It's often linked to diet and obesity, and you get that later on in life. And it's when you still produce insulin, but your body is resistant to your own insulin. So basically it becomes no longer is effective. So you're still producing it, but it's not working anymore. It's not reducing your blood sugars to normal. So your doctor will prescribe, in this case, a carbohydrate controlled diet. Because remembering back from topics on diet, the carbohydrates in paper 1 can be broken down into sugars like glucose. So we need to control the amount of carbohydrates we're taking into our diet, and also he or she will suggest an exercise routine as well to help maintain your weight, which is obviously a risk factor in terms of diabetes. First of all, then, talking about DNA and genes, you need to know that DNA is deoxyribonucleic acid. It looks a little bit like this, in that it is a polymer, which means a long molecule. It is a DNA double helix, and it is found in the nucleus of your cells. Often in the nucleus, if we just take a picture of what the nucleus looks like, often in the nucleus it is just loose DNA because it's being used. It's being used to make proteins. But sometimes it can be organised in chromosomes, and you might see chromosomes like that. Also, you may also see chromosomes that look a bit more like a cross when they have doubled up and got exact copies of each other. So be aware that you might see both of those in the exam. Anyway, in the cell you have 23 pairs of chromosomes, so 46 in total. They're in pairs, they're in pairs because you have one from your mother and one from your father. So you have 46 chromosomes in total. A length of a particular sequence of DNA is called a gene, And that gene is used to make amino acids, of which there are about 20 or so of them, and a particular sequence of amino acids is used to make proteins. Your entire genome, the entire genetic information, in an organism, whether it's a plant or an animal, is called a genome. And that's really important to help us investigate inherited disorders or the things like human evolution and how we're all related to each other and how we've migrated in the past. We are going to draw some more genetic diagrams now, but to be able to do that we need to know a few key terms. The first one is allele. Allele is a form of a gene, or one form of a gene. Try not to say type. A type of gene you will not get the mark for. Say one form of a gene. So a dominant allele means that a characteristic is always expressed if one allele is dominant. So if you have a dominant allele, a dominant form of a gene in your DNA, you will express that particular characteristic. Whereas a recessive allele, the characteristic is only expressed if both alleles are recessive. So what that means is to express something that is recessive, you have to have a recessive allele from both your mother and your father to have the chance of expressing that particular characteristic. A phenotype is what you look like, so it's the characteristic that is expressed. So if they ask you about the phenotype, you might be talking about brown hair, for example, or tongue rolling. It's whatever characteristic you're talking about in the question. A genotype is the alleles present at the genetic level. So your genotype, for example, would be, if we're looking at tongue rolling, it could be T. T, two capital T's. I forgot to mention up here that when we express alleles as letters, a dominant allele is a capital letter and a recessive allele is a lowercase letter. So here we've got TT. This would be called homozygous dominant. Homo meaning the same, we have two of the same dominant alleles. Lowercase t's would be called homozygous recessive. Homo meaning the same, and two recessive alleles because you've got two lowercase letters. And the final one that you can have is one of each, which is heterozygous. So that's going to be important for us to draw our genetic crosses. We'll start here by this example. If you're doing higher tier, you would have to draw the genetic cross from scratch. If you do foundation tier, you'll probably be given the genetic cross and have to complete it. So this question says the ability to roll your tongue is caused by a dominant allele. What is the probability that a woman who is homozygous recessive for tongue rolling has offspring that can roll their tongues if she has children with a man who is heterozygous? So let's use the letter T again. We said recessive would be lowercase t, because homozygous recessive. Heterozygous would be T, T. So we'd cross those in a genetic cross. Capital T, lowercase t, crossed with TT. You'd have to do that from scratch if you're doing higher tier. Foundation, you would have to complete this cross here. So all you do is match up the letters. Here you would get TT. Here you get two lowercase t's, two lowercase t's, and here TT. So if tongue rolling is caused by a dominant allele, anytime you've got a capital letter, you will be a tongue roller. So here we finish it off by saying you've got a 50% chance of tongue rolling. Now we're going to move on to look at inherited disorders, and two that we'll look at are polydactyly and cystic fibrosis. Polydactyly is a disorder caused by a dominant allele, but this allele is quite rare in the population. So even though it's dominant, not many people possess this allele, but if they do pass it to the next generation, then they will be born with an extra digit on their feet or on their hands. This polydactyly is quite easy to deal with. It's dealt with at birth and the extra digit is removed, and lots of people don't even realise that they suffered with that disorder from birth. This other disorder is cystic fibrosis. This is caused by a recessive allele, and this affects the cell membranes. With cystic fibrosis, you can see here a sufferer having lung difficulties. It causes mucus and excess mucus to be present in the airways, in the air passages, because of this disorder with the cell membranes, and what it causes is really severe breathing difficulties, and people find it difficult to get enough oxygen into their lungs, and it can quite severely affect their lives in some cases. So one suggestion to help improve for such inherited disorders is to do something called embryo screening, which is looking at the embryo, which is a ball of cells right at the beginning of life, and selecting a cell from that embryo and looking into its DNA to see if it has genes which could cause some of these genetic disorders. And what they are looking into is perhaps being able to change or remove a certain gene that they find that's causing a disorder or could cause a disorder, or perhaps even suggesting that they wouldn't use this embryo and they would choose another one that the mother and father had produced instead. Obviously, that has a lot of ethical and economical and moral advantages and disadvantages behind it, which I suggest you look further into. Main advantages are that it could eradicate some of these genetic disorders, but obviously there's a lot of debate surrounding it because you're choosing what embryos survive and live, and you're perhaps prejudicing people with these disorders, so there's lots of continuing debate around it. First of all then, we'll look at the idea of competition. And firstly, we'll look at a couple of key words that are important in this topic. The first one is population. And with population, that means all of the organisms of just one species living in a habitat. For example, you could have a population of elephants, or you could have a population of water lilies, for example. Community is the populations of different species living together. So, for example, we might describe a woodland community which comprises of a number of different species, including owls and beetles, etc. Fundamentally, whether you're talking about the individuals within a population or the individuals within a community, there is a lot of competition between those individuals. For example, in plants would compete with each other for things such as light, space, water, and minerals or nutrients that they take up through their roots. Animals, on the other hand, will compete with each other for things such as territory, so similar to plants in the fact that you're talking about space, so territory or space, food, water, and mates. And this links into the evolution topic that we've looked at, whereby those that are best suited for the environment and are the best competitors will then go on to reproduce and pass on their genes to the next generation. Another important word that I want to introduce is the word interdependence. Inter is like between, and dependence dependent on each other. And this is the case where different species are dependent on one another. For example, this bird that is building this nest in this tree is dependent upon the tree to be able to build its nest. Without that tree existing within that community, that bird would have nowhere that it could roost and lay its eggs and bring up its young. So that bird would be dependent on the tree, and we call that interdependence. In this example here, you've got two pollinators pollinating this plant. So that plant is dependent on these two organisms. Another example of interdependence is dependent on them for pollination. And in reverse, the organisms here are dependent on that plant for food. So when you start building up all of the organisms within a community into a food web, you can see with interdependence that any loss of one organism within a community due to disease or predation or something like that causes massive knock-on effects to the whole community. We're now going to look at abiotic and biotic factors. Abiotic factors are anything that is non-living, and biotic factors are living factors. Just like biology is the study of living organisms, bio is anything that is living. So the non-living factors that might affect a community are things such as anything to do with weather really, so the temperature, the rainfall, or maybe humidity or something like that, the light availability, anything really to do with the weather conditions, because they are non-living factors. They could affect all the individuals living within a community. The living factors that could affect or disrupt a community are things such as disease, predators, competition, and food availability. The interaction between the biotic factors, the animals living in the environment, and abiotic factors is called the ecosystem. So the ecosystem is how the animals interact with their whole environment, including the abiotic factors as well. So that's the largest level of organisation you have, much bigger than a community or a population. Next, we're going to look at food chains. It's quite a simple concept, but there are lots of important words involved. This is the idea that we can link organisms in a community depending on what organism eats what. For example, in this food chain we have the grass, which is then eaten by the grasshopper, which is then eaten by the mouse, and which is finally eaten by the owl. So a few key words. First of all, this grass here, and always at the beginning of the food chain you have something called a producer. This is because it produces its own food by a process called photosynthesis. Where that process gets its energy from originally is the sun. So energy is transferred to the plants and they use that energy from the sun to make their own food. That is then consumed by the grasshopper. So all of these three organisms here are called consumers, but this one is the primary consumer because it is the first consumer in the chain. The mouse is what we call the secondary consumer because it is the second organism in the food chain, and the owl is called the tertiary consumer. Now, we don't have a continual line of organisms here. Within a food chain, you only tend to have 4 or 5 or maybe up to half a dozen organisms. After that, it stops. Now the reason for that is because not all of the biomass is passed up the food chain. That means not all of the mass of each organism is passed up each food chain. Because, for example, perhaps the grasshopper can't eat all of the different parts of the plant. Perhaps there's some structures that it can't consume. The mouse, for example, might not eat all of the grasshopper. Maybe it leaves out its wings or its legs because they find those inedible. And the owl, for example, won't digest all of the mouse. It will swallow all of the mouse, but then it will bring up things like its fur and any of the hard bones that it can't digest. So by the time it gets to the end of the food chain, there is not enough energy for another organism. Energy is wasted in other ways as well, for example, through the movement of the organisms, through the fact that they produce feces, and as we said before, the fact that there are inedible, which means they can't be eaten, parts of the organism. So by the time you get to the end, there's just simply not enough energy for another organism to fit into the food chain. That's why the arrows face in this direction. They show the direction of energy transferred. So they always have to be in this direction, moving from the producers through to the consumers. A couple of extra keywords then. We would describe the owl as a predator, and we would describe, for example, the mouse as prey. Anything that's eaten is called prey, and anything that's doing the eating is called the predator. So in the same way, the grasshopper would be the prey for the mouse, and the mouse would be the predator of the grasshopper. Next, we're going to look at a required practical for paper 2, which is all about how organisms are distributed. And this required practical is all about the idea of random sampling of a population. Population being one species, if you remember from the keywords that we looked at before. For example, if you had a very large area, say 100 metres by 100 metres, you might want to estimate the number of a particular organism that is in that area. For example, the one that's often used at school is you might want to measure or estimate the number of daisies in a particular field, and you might want to compare two different fields, sunny versus shady, for example, or different fields with different pHs, perhaps different fields with different grass types as well. There's lots of things that you could look at, but the main thing is that you can't sample the whole field. It would be too time consuming and nigh on impossible to go and calculate the number of daisies in that field. What we can do is we can subsample a much smaller area of the field. It's not to scale, but for example, we could have an area that was 10 metres by 10 metres. And what you do is you divide that grid up again with string. So there we have it. We have a grid in the middle of our field, and we've divided that grid up into 100 squares. So if we were to write a method for this, or start writing a method, we would first of all write that we would measure an area 10 by 10 metres and divide this up into a grid with squares of 1 metre by 1 metre. Then still within that grid we can't sample all of the different squares within there. So what we need to do is we need to use a random number generator, and we need to use that to pick, for example, 10 or 20 squares that we are going to sample. They'll ask you about this in the exam perhaps. The more squares you sample, the more reliable your results are going to be. So we can use a random number generator. So for example, if we assigned these squares from top left to bottom right, 1 to 100, so we'd go along and say that was 1, 2, 3, 4, 5, 6, etc. We could then use our random number generator and say, for example, that it picked out 10 squares at random. We would then sample within those 10 squares, and what would we use is a grid that looks very similar to this called a quadrat. So a quadrat is a metal grid. Sometimes they are 1 meters by 1 meters, it depends on the quadrat you've got. Other quadrats are a bit smaller, perhaps 0.5 meters by 0.5 meters or something like that. So you need to be aware of how big a quadrat you're using. I'm going to suggest we're going to use a 1 meter by 1 meter quadrat, and what we do, we would then place that on one of our sampling squares, and we would count the number of daisies within that quadrat. So we can build up our method here. We've used a random number generator, and now we are going to count the number of daisies in each of the 10 squares by placing a quadrat over the grid square and counting the organisms we find, or counting the daisies in this case that we find. And we would repeat that for each of our 10 squares, and then we would take an average number of daisies per quadrat, and then we would then have an average number of daisies per meters squared. And with that information, you can then compare that to a shady field, repeating it in the same way, measuring an area, dividing it up into a grid, using a random number generator to pick, for example, 10 squares, placing a quadrat on those squares and counting the number of daisies within the quadrat. The second way that you can sample organisms is by using a transect. This is slightly different, although it does use those metal grids that we used before called quadrats. It doesn't have to use quadrats, but it can do. For example, what you might want to do is look at the distribution of organisms in a straight line away from a tree. And what you would do was you'd lay down your measuring tape, which is your transect line, and you would lay that down on the ground away from the tree. Then, at regular intervals, for example, every metre or every 2 metres, you would sample the organisms. And to do that, you could either just count the number of organisms that were touching your tape measure or your piece of string that you put down as your transect line, or you could place your quadrat there and count the number of organisms that you find within your quadrat. Not only could you do that in a horizontal way, you can also do transects vertically, for example, up a tree. You could lay a piece of string or a measuring tape up a tree and sample something like leaf size or lichen cover as you go up the tree. So you might not want to use a quadrat, or you might just want to have a look at regular intervals next to that string line or tape measure. You might want to measure the size of the lichen that you find. In the same way that you can do that on a wall, for example, here you could lay a transect vertically on the wall And next to the transect at regular intervals, you could either place a small quadrat, or you could simply measure the size of the lichen at that point. With a quadrat, not only might you want to count the number of organisms, but for some organisms this is particularly difficult. And instead, you might want to calculate the percentage cover in the quadrat instead. And you may well be asked to do that in the exam. So, for example, if I draw a very, very small, simple quadrat that is 5 squares by 5 squares, in total there are 25 squares. And let's say, for example, that we put this on the wall and we had a particular coverage of lichen, like so. We would count the squares that are covered with lichen. In this case, there are 1, 2, 3, 4, 5 squares that are covered in lichen, and we would use that to calculate the percentage cover. So you do the number of squares with the organism in it divided by the total number of squares, and then multiply by 100 to get your percentage. So in this case, we've got 5 divided by 25 multiplied by 100, and that will give us our percentage cover of 20%. We're now going to look at the water cycle. The water cycle basically explains how on Earth the water is constantly cycled around and reused. So there's some important key words. First of all, evaporation. So this could be water evaporating from the sea, for example. That water would evaporate, and when it gets higher up in the atmosphere, it would cool down and it would condense into clouds. So we've got first evaporation and then condensation. As those clouds move around, at some point they will then turn into rainfall, and this is called precipitation, and that rain will fall back down onto the earth. At this point it might flow, for example, into rivers, straight back into the sea, or it might get taken up by plants, and when plants take up water, that water can then be evaporated from their leaves. So this comes up in paper 1 as well, this process whereby plants take up water and evaporate it from their leaves, and this is called transpiration. So in this process here, through evaporation, condensation, precipitation, flow back into the rivers, and transpiration, that is how the water is cycled on our planet. So a lot of key words there. You might see, for example, the idea that if you have some water in an iceberg, for example, on the sea, they might ask you to suggest how that water ends up in a lake. So here you would have the water, the iceberg melting into the sea, that water evaporating, condensing into clouds, and then you would have the precipitation that would lead to the rainfall filling up the lake. Further to the water cycle, you also need to know how carbon is cycled. So carbon is a compound that you're probably well aware is in the gas carbon dioxide. So carbon dioxide has a major involvement in moving this carbon around the atmosphere, but it doesn't always have to involve carbon dioxide. Let's start over here first of all. Photosynthesis is part of the carbon cycle because plants take in carbon dioxide and then they add that to water that they take up through their roots, and they make a substance called glucose and oxygen. And that you'll see balanced out with 6s in front of the oxygen, the water and the carbon dioxide. So this process, it takes the carbon from the gas here and turns it into cycles it so that it then goes into this molecule here, which is glucose. Animals will then eat the plants, and therefore they will consume the carbon here from this glucose molecule, and that will be taken in by the animal. Animals will then both respire and produce waste, so some of the carbon will be in the waste here, but it'll also be released by the animal as CO2 when the animal respires. Plants also respire, so the respiration from animals and plants will again release CO2 back into the atmosphere. When the animal and the plants die, the carbon that is in these organisms will be recycled through the process of decay. Microorganisms are mainly responsible for decay. So the microorganisms will feed on the dead plants and animals, and they will also respire. It's not just the larger animals and plants that you think of as respiring, but also these microorganisms are respiring, and therefore they are also releasing CO2 back into the atmosphere. Vehicle emissions are also responsible for releasing carbon dioxide. So the carbon starts off first of all locked up in fossil fuels as the hydrocarbons, and then when they are burned through a combustion reaction, that is, as we looked at in the chemistry topic, combustion results in carbon dioxide and water being released, and that is another way that carbon is cycled. And finally, factory emissions. So they are burning fossil fuels, also releasing CO2 into the atmosphere, and this completes the carbon cycle.